Okay, great. Well, hi everyone. My name's Aaron,
And I'm Crystal.
and we both work in a lab at the Harvard School of Public Health in the Manning Lab, and we're excited to take you on a tour of our lab today. So before we get started, we thought we would tell you a little bit about us and our lab. And so I'm a postdoc in the lab, which means that I've been to grad school already where I conducted a research project. And now I've moved to this lab where I'm doing a new project that is sort of independent research. And Crystal is a grad student within this lab. And so, yeah, we're excited to tell you about what we work on. And our lab is really interested in understanding how all of the cells that make up our body interact with the environment around them. So how they take up nutrients from around them, how they detect different signals, and how this allows the cells to do all the different things that cells need to do, grow, and other functions. And so we're mainly interested in the different regulatory processes that occur within cells. Our lab, specifically focuses on the nutrient side of things. So we're interested in understanding how metabolism works within your whole body. We're also interested in understanding how individual cells take up nutrients, what they eat, and what they do with these nutrients that allows cells to do different things. We're also interested in how cancer cells grow very rapidly and what sort of, how the different processes that allow cells to regulate themselves are used by cancer cells to grow very quickly. And, so the hope is ultimately to be able to better understand diseases in humans and also sort of how humans in a healthy state, how we work basically. So yeah, I think, I guess we can get started on the tour.
Definitely. So I'm going to start off by showing you guys how we work with cells and how we study cells in order to learn how they function and also how they grow. So follow me.
Okay.
I'm taking you guys to see our cell culture or tissue culture room. And you can see that we have two of these really large hoods in the room. And this is a really protected and sterile environment in which we have to work with our cells in order to know that the cells that we're working with are really specific and pure and that we don't contaminate them with any bacteria that's in the air or on our hands. So first I'm going to grab some gloves. So some of the cells that we work with are cancer cells. So we can try to understand what's going wrong with them and what's making them grow so fast. And then we also work with regular normal human cell lines too to understand the processes in healthy tissue. So open up the hood. And then I have to spray all the inside with alcohol, which helps to just sterilize and remove any germs that can contaminate our cell culture. And then here's where we store our cells, and it's called an incubator, and it keeps the cells at a specific temperature that helps them grow and stay healthy, which is 37 degrees Celsius. So we have our cells in a lot of different containers. So we have these dishes and we have these multi-well plates where we can do experiments. So here is one plate. So that was an awesome question. Yeah, so I guess everything that we do, we want to make sure that we're keeping the cells really safe and in the best environment where they're not going to be contaminated, they can grow. So, so I have my plate of cells here, and again, if I wanted to work with them, what I have to do is something that's called like an aseptic or sterile technique, where you want to make sure that whatever is going to touch your cells or the liquid that your cells are growing in, which gives them nutrients and everything that they need to grow. You want to make sure that whatever's touching the cells doesn't touch anything else. So we have these sterile, what's called pipettes, which helps me bring up media or the liquid that feeds the cells. And it's sterile here. So I just open the pipette and I would attach it to this that allows me to draw up or dispel liquid. And I'm going to make sure it's not touching anything else. And I can take the lid off and just bring liquid up or re-pipette down. And that's just like part of the technique that we use to make sure that nothing gets contaminated. But normally you would have like a bottle of media here, or if you wanted to transfer the cells, you could do it like that. So basically what we do with the cells, we can modify their genes and look at what effect a certain gene has, or we can modify the type of food that we give them and see how that changes their growth and things like that.
Why are so many cells in the body? Do you want to do this one or?
Well, our entire body, like you said, is made up of cells. And the reason why there's so many is because they're so small and they all have really specific functions. So we have certain cells that make up our liver, certain cells that make up our skin, and all of our different organs have very specialized cell types. And so it takes a lot of cells to keep each organ running and each tissue running, and then it takes a lot to just keep our entire body functioning properly. So yeah, if you think about how big you are and how small a cell is that we have to even look at it under a microscope, then you can imagine how many of those small cells it's going to take to make up our whole body.
Okay, great. So now we can move on from tissue culture.
Okay. So another thing that we are interested in looking at in our cells are proteins. And so proteins are kind of the molecule in our cell that does all of the functions and carries out all of the work that our cell needs to do. So you've heard of DNA probably, which is, we know, is the genetic material that provides all the information in the cells in our body to tell them what needs to be done and what to do. And so what happens is the DNA gets kind of made into a messenger and then made into the proteins and that carry out all the functions in the cell. And so sometimes we want to look at how much of a certain protein is expressed, how much of a certain protein there is in our cells. And that might be because there's so many different proteins in our cells that carry out so many different processes. So if we want to look at a specific protein, we can use what's called a technique called a Western blot. So Western blot is a technique where we can apply our proteins to the gel, and then the gel will separate the proteins in our sample based on size. And then we can image them on something like a film, and we see these bands showing up, and that tells us how much protein there is. So if you have like a lighter image, there's less protein, and then if you have a darker image, there's more protein. Yeah, so are there any questions about proteins and what different kinds of proteins there are, what they do?
How do we get the nutrients to feed a cell?
So basically we use a liquid that we call media, and it contains all the nutrients, like you said, that our cell needs to grow. And I guess we buy that, and it's based off of, I guess, what your cell might see in your body. It contains similar nutrients to what the cell might see in your body, but it's just made in a container.
Yeah.
Okay. Okay, so now we're gonna switch to Erin. Erin is gonna be showing us some more that we do with proteins.
Great. So yeah, Crystal talked about using Western blots to understand how much of different types of proteins we have within a cell. And now I'm going to take you to a machine that we use to purify different proteins. We want to extract them inside a machine that we call an FPLC, which is an acronym that basically describes a machine that is trying to purify proteins. So sometimes we're not interested in just understanding how much of a protein is in a cell. But we want to be able to purify that, and then we can study this protein once it's been purified. And so this is the machine, and it uses a lot of different pumps and liquids and these columns in order to purify proteins. So we can take a sample that has a lot of different proteins in it and inject it into this machine. And when it's in this machine, it flows through these columns, and these columns will separate the proteins on the basis of their size. So we can collect small proteins, and we can collect big proteins as well. And by doing this, potentially several times, we're able to really isolate individual proteins if we want to study them. And so once the proteins flow through these columns, they actually go into this part of the machine, which is called a fraction collector. And so here we have these tubes that will contain individual proteins within them. And so once we purify the proteins in these tubes, we can study their functions in different experiments. And just to also show you, we have a computer that goes with this, and we get data on the proteins and where, which fractions they'll end up in. And this tells us something about how big the proteins are that we're really interested in studying. Yeah, so are there any questions about this? So for this kind of experiment where we're trying to understand the protein size or other information about it, information like the size could be some data that we obtained from this experiment. In the previous experiment that Crystal showed, where we are trying to understand how much of a protein, of a certain type of protein is inside a cell, then the amount of that protein is a form of data that we collect and understand. So, data can be a lot of different types of things, but it's basically any information that we're trying to learn when we do a scientific experiment.
Great.
So, after we've talked about the amount of proteins that we have in cells, we can also talk about how we understand purified individual proteins. But something we're also very interested in understanding is what these proteins are doing inside a cell, where they are in the cell, and sort of if different things are changing the position of these proteins inside a cell. And so to look at this, we use microscopy techniques to really zoom in on individual cells and get an understanding of where different proteins are inside the cell. So now we'll take you to our microscope where we can show you some examples of what we look for when we're looking at cells. Okay, so we keep our microscope in this room over here, and this is our microscope. It's a sort of very large setup that allows us to take a look at what's going on inside a cell. And so normally when we're using this, we look through these eyepieces, and then we can zoom in and see very, very small things. And so we can see cells that are placed on this stage. We also have this machine equipped with some lasers, and the lasers allow us to look not only at the whole cell, but we can look for certain colored molecules. And why this is very helpful to us is if we're trying to study an individual protein inside the cell, we can label that protein with a green dye or a red dye or a blue dye, and then we can look through the microscope to see where these different colors end up within the cell. And so by doing that, we can not only look at the whole cell, but we can look at where specific proteins are inside the cell. And so I wanted to show you an example of how this type of experiment can look. And so I pulled up on this computer a picture of some cells. So these are some pictures of cells that I took recently, and this is a very, very zoomed in image. So over here, you can see here's a cell, and so we have these individual cells that have been imaged. And we've labeled the cells with different colors. So we have blue, we have green, and we have red as well. And so by studying where these are inside the cell, we can get a better understanding of what is happening to the different proteins in the cell. So for example, here we've labeled the cell's DNA in blue. And so Crystal mentioned earlier that DNA is the hereditary material in the cell. And so that's kept within the cell's nucleus, which is why you see the blue as a big circle kept within the middle of the cell. And then I've labeled two other proteins, one in green and one in red. And you can see those form little dot structures all throughout the cell. And I'm really interested in understanding what happens to the positions of these dots under different conditions. And something that's also very interesting to us is understanding where the patterns of these two colors overlap. And so you can see there are a few spots here that are colored yellow, which is to indicate the overlap of the red and the green signal as well. And so this is just one example. We use microscopy to stain a wide variety of different cell types. So here I'm looking at one type, but there's another postdoc within our lab who looks at neurons, which are the cells of the brain. And she can do similar experiments there to get a sense of how different proteins are regulated or function in those types of cells. Great, so I think that's all I wanted to show about microscopy. Yeah, are there any questions? Great, well, yeah, I guess if there's no questions, we can, I guess, wrap up. So yeah, thank you so much for coming on into our lab with us today. I think we've shown you a number of different ways that we study how cells work and how different proteins that make up a cell work. So we can manipulate cells in cell culture. We can study the amount of different types of proteins using a Western blot. We can purify proteins on the FPLC back in the hallway. And we can look at the position or localization of proteins inside of a cell using microscopy. And so all of these techniques allow us to better understand how cells detect different nutrients and materials and signals outside of them, and how this changes the different behaviors that cells have. And yeah, so thank you for coming on this tour with us today.